This video is to show the procedure for the selective and differential media lab that you will be performing as one of the activities from your Carolina biological kit you received. Before you begin, make sure you have these necessary supplies. So you need an activated broth of micrococcus. This should have been saved from the previous lab since we use micrococcus. And you're also going to need E. coli and staphylococcus. These you have not yet activated, so you'll have to follow the procedures from previous labs to activate and incubate E. coli and staph to the point where you can see growth or turbidity in each of those activated broths. It's only at that time when you have 3 grown bacterial cultures that you can do this lab. So make sure you have activated grown cultures of each of those 3. And then you're going to need inoculating loops and candles, since you're going to be opening and closing glass tubes. We're going to be flaming the mouths of those tubes. And the 3 different types of media that you're going to be inoculating are McConkey agar plates, mannitol salt agar plates, and the EMB agar plates. So there are going to be 3 different plates that you're going to be incubating. So I want to show you the media. So this is the mannitol salt agar plate. This is the McConkey agar plate. And the third is the EMB plate. And notice how each of them I've subdivided into 3 regions and labeled with each of the 3 bacteria. So I'm going to focus on just one plate at a time. So I'm going to start with the mannitol salt agar plate. And so I'm going to get my candle lit so that I can do the flaming of the tubes. So there are 3 bacteria that I'm going to inoculate onto each plate. And so again, I've labeled the bottom of the plate. So the first is E. coli. So of the 3 bacteria, E. coli is one of the 3. And so this is a reactivated culture of E. coli that I've been growing to the point where I can see turbidity, so I know there's bacteria. So I'm going to hold on to that lid and open it with my pinky finger, flame the tube, and get a loop full of E. coli that I'm going to inoculate onto just 1/3 of this plate. So again, I've subdivided the plate, so I'm going to make sure I put this on the E. coli section, and I'm just going to streak in a zigzag fashion and then discard my used loop into isopropyl alcohol. Then using a fresh loop, so a sterile loop, I can do the other 2 bacteria. So the other 2 bacteria are micrococcus and staphylococcus. So this one's micrococcus. So I'm going to do this in the exact same way. I'm going to open the tube and hold on to that lid, flame the tube, and get a loop full of micrococcus. Again, this is grown to the point where I can see turbidity. So flame the tube before I close it. And then I'm going to streak this into the micrococcus section. So don't touch it to the E. coli. These are separate sections. And then discard that loop. And I have one more bacteria to put on, and that's going to be the staph culture. So this is a fresh sterile loop. Same procedure, loosen the lid. I almost forgot to flame it there. So flame that mouth of the tube and then inoculate or grab a loop full. And then I'm going to inoculate the final section, which is labeled as staphylococcus. So this is one of the 3 plates that I'm going to do. And of course, there are 2 more plates that I'm going to do in the exact same way. So this next plate is the EMB plate. So it too has been labeled into 3 separate sections that I'm going to do the exact same procedure. So add each of the 3 bacteria. So flame the tube, grab some E. coli. I should have flamed that tube before I closed it. And I'm going to add the bacteria to the E. coli only section, and of course discard the used loop. Now I have 2 more bacteria, so get a fresh loop and then grab the next bacteria. So this is going to be the micrococcus, so flame that, grab a loop full, and again using aseptic technique, flame the tube and close it. And then this is going to be added to the micrococcal section. Discard the loop. Grab a fresh loop for the 3rd and final bacteria for this plate. So this is staphylococcus. So flame the tube, get a loop full of inoculum, flame the tube and cap it. And then of course, inoculate the plate in the staphylococcal region. And that completes that second plate. That's the EMB plate. And the final plate is the McConkey agar. So we're going to, again, repeat the procedure. So starting with a loop full of E. coli, I'm going to grab that and then, of course, flame the tube prior to closing. So that's the last time I'll use the E. coli in this experiment. So inoculate the plate with E. coli, discard the loop, grab a fresh loop. This is the micrococcal culture. So flame the tube, grab a loop full of inoculum of micrococcus, flame the tube prior to closing it, put that down and inoculate the second region, which is micrococcus. And the third and final bacteria is staphylococcus. So same procedure. Again, just double check you're putting the bacteria in the proper section on the plate. So when you see growth, you can be confident that it is the bacteria because you double and triple check that that was the area. So I don't know if you notice what I do is I put my finger on the zone before I pick it up. So when I flip the plate over, I can tell what section I should inoculate onto. So the next steps include making sure that all of these tools remain in the disinfectant for the full 30 minutes, as we've done in each and every lab. In terms of cultures you need to save, all three of these bacteria that you just used, the broths that you have, so E. coli, staphylococcus, and micrococcus, we need in the next lab activity. So do not discard any of these three broths. You'll need them for the next activity. And then these plates need to incubate, like all of our plates, we do so upside down. And you're going to be observing these plates after 2 days, so 48 hours, again at 72 hours, and then a third time at 96 hours. At each of these time points, what you want to do is make and record your observations in the assignment sheet. So there's a data table where you're supposed to be looking for different results. And then, of course, taking photos at each of these time points as well to demonstrate that you've completed this lab activity appropriately. I'm going to make an additional video to talk about the results of each of these plates to make sure you understand what the results that you see mean and how you can successfully interpret each of your plates.